Hello everyone, welcome to my YouTube channel. I hope you all are fine. We are discussing BRS Physiology Chapter 1, and in this video we will talk about neurotransmitters. So I have made this video in a different way for a better understanding. So let's get started. Okay, so we have two types of neurotransmitters, the excitatory one and the inhibitory ones. So the excitatory neurotransmitters include acetylcholine, norepinephrine, epinephrine, dopamine, serotonin, histamine, and glutamate. Whereas the inhibitory neurotransmitters include GABA, that is gamma aminobutyric acid, glycine, and nitric oxide. Okay, so let's discuss each of these neurotransmitters individually. Okay, so the first excitatory neurotransmitter, that is norepinephrine. Acetylcholine, we have talked about acetylcholine so much in the previous video, the Neuromuscular Junction. If you haven't watched this video, go check it out first. I will add the link to that video in the description. Okay, so norepinephrine. Norepinephrine is released from the postganglionic sympathetic neurons. Okay, so the sympathetic and parasympathetic nervous system, we will talk about it in the next chapter in detail. Okay, so remember that we have preganglionic neurons and the postganglionic neurons for both sympathetic and parasympathetic nervous system. And the preganglionic neurons, in case of parasympathetic nervous system and the sympathetic nervous system, they will release acetylcholine. So the preganglionic nerve fibers for both sympathetic and parasympathetic nervous system, they will be cholinergic because they release acetylcholine. But the postganglionic sympathetic neurons, they release norepinephrine. So this norepinephrine, it binds with the alpha and beta receptors on the postsynaptic membrane. So the target cells in the body, they have the alpha and beta receptors to which the norepinephrine, it binds. And how it is removed from the synapse? It is removed from the synapse by reuptake, or it is metabolized by two enzymes. One is the monoamine oxidase and another is the catechol orthomethyltransferase. And it is metabolized into various metabolites like 3,4-dihydroxymandelic acid, and the name of these metabolites are written in BRS Physiology. You don't need to remember all of them. And then there is this disease, pheochromocytoma. So it is a tumor of adrenal medulla, and it results in the increased secretion of catecholamines like epinephrine and norepinephrine. And this results in the increased urinary excretion of VMA. So VMA is one of the metabolite of norepinephrine, and that is the vanillyl mandelic acid. Okay, so next we have epinephrine, and it is synthesized from norepinephrine by the action of an enzyme that is a very long name for this enzyme. So it is a phenylethanolamine N-methyltransferase in the adrenal medulla. Okay, and the next we have is dopamine. So dopamine is a prominent neurotransmitter in the midbrain neurons. And when it is released from the hypothalamus, it inhibits prolactin secretion. So that is why it is also called as a prolactin inhibiting factor. And it is metabolized by monoamine oxidase and catechol orthomethyltransferase, these two enzymes like we talked about for norepinephrine. Okay, now let's talk about the dopamine receptors. The dopamine receptors are G protein coupled receptors. And what are these G protein coupled receptors? So the G protein coupled receptors are integral membrane proteins, and they are involved in transmitting the signals from a variety of stimuli outside the cell to its interior. Okay, so these dopamine receptors are of two types. We have D1-like receptors, which include D1 and D5, and they are linked to the G protein that is stimulatory in action, that is the GS protein. This will activate the adenyl cyclase and resulting in the increased concentration of cyclic AMP. And we also have the D2-like receptors, which include D2, D3, and D4, and they are linked to the G protein that is inhibitory in action, the GI protein. This will inhibit the adenyl cyclase, resulting in the decreased concentration of cyclic AMP. Also activates the potassium channels and inhibits the calcium channels. Okay, then we have two diseases related to dopamine. One is the Parkinson disease. There is degeneration of dopaminergic neurons that use D2 receptors. So in Parkinson's disease, there is decreased dopaminergic activity. And second one is a schizophrenia, in which there is increased level of D2 receptors. So in schizophrenia, there is increased dopaminergic activity in the body. Okay, so we talked about dopamine, norepinephrine, and epinephrine. Now there is a synthetic pathway for these three, and these three are formed initially from the amino acid tyrosine. And this tyrosine is converted into L-dopa by the action of the enzyme tyrosine hydroxylase. Now this L-dopa is converted into dopamine by using an enzyme dopa decarboxylase. Now this dopamine is converted into norepinephrine by using dopamine beta hydroxylase. And now remember that epinephrine, it is formed from norepinephrine by an enzyme phenylethanolamine and methyltransferase. So this is the synthetic pathway for these three. And remember that tyrosine is the amino acid that is initially used for the synthesis of these three neurotransmitters. And then we have three other excitatory neurotransmitters. So serotonin, it is present in high concentration in the brain stem, and it is formed from the amino acid tryptophan. And the next is the histamine. So it is present in the neurons of the hypothalamus, and it is formed from the amino acid histidine. And the last one we have here is the glutamate. So if someone asks you about the most prevalent excitatory neurotransmitter in the brain, that is glutamate. So glutamate is the most prevalent excitatory neurotransmitter in the brain. And glutamate receptors, there are 4 subtypes of glutamate receptors. The 3 subtypes are inotropic receptors, and these are the ion channel associated receptors. One example is this NMDA receptor, that is N-methyl D-aspartate receptor. And the one subtype is the metabotropic receptor. So this is a G protein coupled receptor because it is coupled to ion channel via a heterotrimetic G protein. Okay, now let's talk about the inhibitory neurotransmitters. So we have 3. One is GABA, and glycine, and nitric oxide. So GABA, that is gamma aminobutyric acid, it is an inhibitory neurotransmitter. It is synthesized from glutamate by glutamate decarboxylase. 2 types of receptors we have for GABA. One is the GABA A receptor. This is an ion channel associated receptor, increases the chloride conductance. And what happens that it results in hyperpolarization and thus decreasing the occurrence of the action potential. And this is the site of action of major drugs like benzodiazepines and barbiturates. And the second receptor, this is GABA B receptor. It is a G protein coupled receptor. This increases the potassium conductance, decreasing the calcium conductance, and this will decreases the concentration of the cyclic AMP. Okay, glycine, this is another inhibitory neurotransmitter that is found primarily in the spinal cord and brain stem, and it also increases the chloride conductance. And the last inhibitory neurotransmitter is the nitric oxide. So it is the inhibitory neurotransmitter in the GIT, blood vessels, and the central nervous system. How it is synthesized? It is synthesized in the presynaptic nerve terminals, where this enzyme, anosynthase, converts arginine into citrulline and nitric oxide. This nitric oxide, actually, it is a permeant gas that diffuses from the presynaptic terminal to its target cell, and it functions in signal transduction using guanylyl cyclase in a variety of tissue, including the vascular smooth muscles, where it causes vasodilation. It also functions in neurotransmission, apoptosis, and tumor growth. Okay guys, that's it for today. Thank you so much for watching. Like, share, and subscribe to my channel. I'll see you in the next video.